Welcome. Welcome to our third Longwood Seminar of 2016. I'm Gina Vild. I'm the Associate Dean for Communications and External Relations at Harvard Medical School. We're going to have a full house tonight, so may I ask you to please move into the center so people who arrive late can take the end seats and it doesn't disturb the speakers. Thank you so much. I'm happy to report that as a result of your attendance, both through live streaming and in person, this has been the most successful mini med school we have had to date. So I want to thank you. Your engagement is impressive and it is certainly appreciated. Your completing the evaluation forms helps us with our planning going forward. I encourage you to please complete the form tonight. That will guide us as we go into the next year. Your help in crowdsourcing the topics helped ensure these were both timely and interesting to the community. And your engagement in social media, Facebook, Twitter, and Instagram has really helped expand knowledge to the public. So I invite you to join our conversation tonight on Twitter with hashtag HMSMiniMed. I hope you'll join us for our last Mini-Med School Longwood Seminar on April 22nd. It's the new old age. And we will learn all about how our body ages, and importantly, we'll learn how to keep it young. Certificates of completion are available next week if you've attended 3 or more mini-med schools. If you can't be here, please send us your email address through at the email on the screen, and we'll mail it out for you. Now, tonight's topic, the seminar that you've all been waiting for, is Taking It All In: Environmental Toxins in Your Health. Rachel Carson, who you may know, she wrote in Silent Spring, one of the landmark books of the 20th century. If we are going to live so intimately with chemicals, eating and drinking them, taking them into the very marrow of our bones, we had better know something about their nature and their power. In 1962, with this book, she sparked the modern environmental movement. Silent Spring introduced new ideas that rapidly became fodder for public discussion. People began talking about the effect of toxic chemicals, especially DDT. Their effect on wildlife, our environment, and on human health began to be questioned for the first time. We've learned a great deal over the past 55 years about the interconnection of nature and human health, yet thousands of everyday products are made with chemicals: clothing, cosmetics, toys, food containers, to name just a few. And we know that many of the chemicals are not adequately regulated. Rachel Carson also wrote, if we have concluded that we are being asked to take senseless and frightening risks, we should look about and see what other course is open to us. And that is exactly what we're going to do tonight. Tonight, Harvard scientists who are experts on the nature and power of some common environmental toxins will help us see what other course may be open to us. So let me introduce them. Monica Koliakovo, who is a professor in the Department of Genetics at Harvard Medical School. Moitrey Muzamdar is an assistant professor of neurology at Harvard Medical School, assistant professor in environmental health at the Harvard T.H. Chan School of Public Health, and a pediatric neurologist at Boston Children's Hospital. But first you'll hear from David Christiani, a professor of medicine at Harvard Medical School. He is the Elkin Blount Professor of Environmental Genetics and the director of the Harvard Education and Research Center at the Harvard T.H. Chan School of Public Health. He is also a physician in the Division of Pulmonology and Critical Care Medicine at Massachusetts General Hospital. Dr. Christiani has examined genetic factors that make people susceptible to lung cancer. At Mass General, he also researches environmental conditions of the lung. He runs a landmark 35-year study of respiratory diseases that has determined the rate of loss in lung function among dust-exposed workers. In 2012, President Barack Obama appointed him to serve on the National Cancer Advisory Board. Please welcome our guest speakers. Thank you.
Thank you all. Let's see. We're going to load this up. There we go. Thank you all. Good evening. And it's wonderful to see the big turnout we have tonight. I'm going to use environmental cancer as an example of environmental conditions that are preventable, so they're caused by the environment or to the extent that we know the environment contributes to their causes, which is a problem, but it's also a situation where there's the opportunity for prevention. And so the title is Preventing Environmental Causes of Cancer. There are only a few slides that are technical, and I'm going to reduce them very quickly to very understandable English. So I'm going to cover some basic landmarks of what we call cancer epidemiology, the study of disease distribution and risk in human populations, what's known about exposure in cancer, why do we even make this link, and then I'm going to touch on a presidential report that had implications for global health, cancer health. The report was focused on chemicals in cancer, and then talk a little bit about the challenges that we face in the 21st century. So what are the landmarks in our understanding about how cancer causes, how environment causes cancer in human beings? In the 19th century, cancer was somewhat unusual because people didn't live that long, and when it occurred, it was generally considered incurable. 20th century, fast forward to about mid part of the 20th century, much of human epidemiology, particularly in Europe and North America, shifted from the study of infectious diseases, flu, pneumonia, diarrhea, to chronic disease, i.e. now cancer. And then after World War II, cardiovascular disease. So you can see this epidemiologic shift in what causes morbidity and mortality in countries across the world as we move from infectious causes of death and perinatal and childhood respiratory infection and diarrhea to chronic disease. Well, cancer is one of those chronic diseases. So exposures to tobacco, diet, and some environmental chemicals. So that was about the time when researchers first entertained the possibility that cancer can be prevented because they understand the cause. You can't prevent something you don't understand the cause of, right? So then late 20th century, early 21st century, where we are now, epidemiologic studies have generated population-based data identifying risk factors for cancer that may have multiple risk factors, not a single risk factor, and the development of new technologies to identify biological molecules of exposure, to identify genetic susceptibility, and creating deeper understanding of what causes it, and perhaps also more opportunities for prevention. And that's where we are today. Now, cancer across the world varies quite a bit. This is a busy slide, but all you need to know is the colors. The red areas are very high in certain cancers, and the green areas for the same cancers are low. So in the case of lung cancer, the red areas, cervical and stomach cancer, is very high in certain parts of the world where there's not refrigeration. Lung cancer is very high in the developed countries or industrialized countries of the world, and those undergoing rapid development like China. So any kind of color map you use of cancer incidence varies a lot according to exposures and the economic status of the country, which helps determine those exposures. Breast and prostate cancer, for example, are much more common in people in the industrialized world. And then if you look here in the US, the number one cause of cancer deaths in both men and women remains lung cancer. It's starting to trend downward because of smoking cessation efforts that started as early as 1964, when you see there was still a steep rise, but real serious attempts to restrict smoking that came into place in the 70s and 80s, where it was banned in workplaces, public places, as you all know, hotels, airplanes, et cetera. Then you start to see exposure patterns, use patterns of cigarettes decline. And then there's a lag period for a population to get sick from environmental cancer usually is about a 20-year lag period from when an exposure is introduced into the population. So for cigarette smoking in Great Britain, 20 years after World War I, you see this peak. U.S., it was around World War II, where there's a big bump in cigarette smoking and when women started to smoke more, and then you see 20 years later. Well, it happens with the other side, too. When you start to see preventive efforts be put into place, it takes a while for the incidence to turn around and start to come down, and which is what you see there. This is women, where unfortunately lung cancer kills more women every year than the combination of breast, ovary, and colon. And so it's still, and will be the #1 killer for a long time in both genders in the US. Then I also want to touch on environmental cancer. So the reason I talk about smoking, by the way, is there was a strong environmental cause of cancer, of lung cancer. We know what the cause is, even without teasing out all the chemicals in lung cancer, and I'll come back to that in a minute. But you know that stopping or cessation efforts leads to a decrease in incidence. So we're able to, by identifying and intervening, make a difference. Now, childhood cancer, there's been a lot of debate about whether childhood cancer is increasing in the US. It's the second commonest cause of death of children in the industrialized world, and about 10 to 11,000 kids were diagnosed with cancer probably pretty much every year, and almost 1,600 will die. So if you look at, and this is just look at the line, if you look at the surveillance data on children with cancer and do all the different statistical adjustments over the decades of 75 to 2008, there has been a gradual increase in US mortality for all childhood cancers under the age of 20. And we don't know why. We suspect environment probably plays a role. This is just a pie chart of the kinds of cancers that children get. By far, leukemia and brain or central nervous system tumors lead the pack, with a variety of very rare, much rarer ones in there. So what causes cancer? Why do we focus on the environment? Well, there are a number of external factors that give us opportunities to intervene in cancer prevention. So one is industrial exposures. Almost everyone in the room has probably heard of asbestos and the problem that asbestos caused and has caused and still is causing in parts of the world, including the industrialized world. But there are many others. High exposures tended to occur in workplaces because of the handling of the material, less so in the general environment. But you can have exposures to cancer-causing agents in water, soil, such as contamination with arsenic or other carcinogens. Then, of course, there are lifestyle exposures. They're just as environmental, but they involve personal choice, whereas in workplace would be an involuntary exposure, or in the general community. Tobacco use, diet, physical inactivity, and in the case of viruses, sexual activity can transmit carcinogens or cancer-causing agents that can then gain access to the body. Naturally occurring exposures, and we should point out that not everything natural is safe. So ionizing radiation, ultraviolet light from the sun, radon, arsenic, infectious agents, they're natural, but they're also dangerous. Medical treatments, not to be underestimated these days. Chemotherapy, which cures a lot of people every year, but the agents themselves are carcinogens, and they put treated people at risk for second tumors. Radiation, immune-suppressing drugs, all can be actually, they're all external causes of cancer. Now, there are also some internal factors that are not easy to control, if at all. Genetic alterations, hormone deficiencies or imbalances, immune deficiency. And some individuals are more susceptible to cancer than others based on heritable or heritable genetic traits we get from our parents, or sometimes of rare mutations that we don't know why they happen. And there's no other way to talk about that other than bad luck. Or what we call comorbidities. If you have one disease, it's not cancer, but it's an inflammatory disease, like inflammatory bowel disease, that inflammation over time can put you at risk of cancer. So you have one condition that's non-cancer contributing to cancer development, so-called comorbidities. So let's talk a little bit about the history of exposures. The occupational exposures were the most dramatic carcinogen experiences for human beings, with with reference to it dating back to the Renaissance, but the first known description was by a Dr. Percival Pott in London, who described an unusual form of cancer of the scrotum of young boys who were chimney sweeps. And we've all read Charles Dickens. You suffered through Dickens in your freshman year of high school. You know that the conditions of the working class in Britain in the 18th, 19th century was very bad. These kids were small, and they were up in the chimneys sweeping out coal, soot. And the soot he found, he didn't know what was in it that caused cancer. He had no idea, but he just made the connection between soot and getting into the folds of the skin of the scrotum, and then taught the kids how to wash, and they stopped getting this kind of cancer. So that was the first, every toxicology course on cancer talks about the first connection made between a definite external agent and cancer in the literature. But it took another 130 years before it was identified what it was in the soot, the chemicals, the polycyclic aromatic hydrocarbons that cause the cancer. In the 20th century, we had many other examples because of the rise of industry. Asbestos, benzene, coke ovens, things called amines, aromatic amines, dyes, etc. And still today, more in the industrializing world, that occupational exposures probably cause at least 6% of cancers in the developed countries and much more exposure in the developing world. And so what Pott didn't know about in the soot that they found out in the 1930s and 20s was there's this group of compounds that's also in tobacco smoke, it's in diesel exhaust, it's in oil emissions and coal emissions, and they're called polycyclics or PAHs. And we now know that those are a very active bunch of compounds, they're not single one, that cause cancers, and not just lung cancer, not just skin or scrotal cancer, but also bladder and gut cancer. And they're in our everyday environment, not just in the chimneys of London or in the workplaces where coal and oil is burned. So urban air pollution is loaded. Wherever combustion takes place, there's basically polycyclics released. There's a steel plant in China that's obviously a polluted workplace, but these are dormitories and buildings where people live right here. And so it's a very intense experience for them to live and work in this steel industry. And a lot of these are polycyclic aromatic hydrocarbons that are cancer-rich compounds. Cigarette smoke has it, plenty of them. And this tobacco smoke carcinogens themselves are a mixed group and can cause a series of reactions that can lead to damage to DNA and then mutation and then cancers like lung or bladder cancer. We looked at this in a group of patients at Mass General as to why young people are at higher risk of, young people who start smoking at a younger age are at higher risk of getting cancer later in life than old people who start at an older age, and found that there are more of these PAH bound to DNA in individuals' lung tissue if they started smoking at a younger age than if they started smoking at an older age. So it looks like it's permanent damage in the lung tissue that carries through later in life. And we found that the people with certain kinds of genetic variation in metabolism are more susceptible to form these so-called adducts, which are bonds, chemical bonds to DNA in lung tissue. Finally, we also found when we looked at blood versus lung, again, this is a pretty good correlation line that the circulating blood was a pretty good substitute, because we can't get lung tissue on people in population studies, was a pretty good substitute for lung in measuring these things. So urban industrial air pollution, mainly of air, but also of water, are rich in these kinds of compounds. We also, as another example, took a very different tack, looked at leukemia in children in Taiwan. Again, it's a busy slide, but I'm just going to point to this line right here. When we looked at children with leukemia, their risk of having leukemia, childhood leukemia, was about 54% higher if they lived within 1 kilometer of a petrochemical refinery than kids who lived farther than 1 kilometer, adjusting for all other factors. So I want to touch on the cancer panel just for the last couple of minutes. The panel was established in 1996 during Bill Clinton's reign to address, frankly, as possible key issues in cancer. And each panel is done by topic, and there are several experts who then go around the country, interview experts, and talk to people in the field and talked to community members about what concerns them about cancer. And they did a couple of reports. One was on lifestyle factors associated with cancer, such as tobacco. The other was diet. And the third was inactivity. And in 2006, the panel was asked to talk about chemicals and cancer, realizing that these other issues had been dealt with. And so a committee was convened, and they reported a fairly hard-hitting report that generated some controversy because the committee was shocked when it found and reported that 80,000 chemicals were on the market and only several hundred had been tested, pre-tested for their cancer potential. Since the early 70s, the International Agency for Research on Cancer had evaluated some 900 suspected carcinogens and found that 165 turned out to be definite in human studies, and another 265 is possible. And so this is a small group of chemicals compared to what's on the market. And so they criticized the current regulatory approach in the US as evaluating only when there's evidence of possible danger and setting standards only when there's evidence of severe health effects rather than a precautionary or preventive approach. So they wrote a strong letter to the president saying there's a growing body of evidence linking environmental exposures to cancer, and the public needs to be, is becoming more aware and more concerned, is expressing the concern, and that we need to do more about it. The panel highlighted a number of things individuals can do to reduce their exposure to potential carcinogens. This generated some controversy because a lot of them had not been tested in sort of trials, although they are prudent, except for number 5, which you all know, eliminating exposure to secondhand smoke is important. So then the whole issue of lifestyle versus environmental risks come up. It's impossible to quantify these things. They're both very important, so that some 45% of cancer in the West is felt to be due to either smoking, diet, or lack of exercise, but that leaves the other 55% that we need to grapple with. How much of that is environment is still up for grabs, except we know that probably 7 to 19% of that 55% is due to environmental exposures. The thing to be careful with all these percentages is sometimes they interact. You can have a lifestyle factor and an environmental factor adding up to bigger problems, such as asbestos, like in this picture, and asbestos and smoking together, rather than simply add risk to 15, multiplies to risks of greater than 50. So we have to do a lot to adjust our environments to minimize exposures to potential carcinogens. And some colleagues have raised the concept of the exposome. We have the genome, we have metabolome, we have the exposome, life course exposure starting prenatally throughout life. What can we do to minimize exposures to harmful agents that cause conditions like cancer and others? So the challenge in our studies are to do better exposure assessment, where we can actually, in our population studies, figure out what we're exposed to, when, and for how long, so we can get better estimates in our studies. So global cancer epidemiology, this is what I want to end on. We should keep in mind that more than half of new cancer cases and deaths each year now occur in low to moderate income countries. It's no longer just a disease of industrialized nations. As infectious disease comes under control, you see more chronic illness like cancer and cardiovascular disease causing mortality in the world. And the spread of a so-called Western lifestyle is an important factor, but also the exposure potential for industrial chemicals like asbestos, benzene, and silica. So we don't want to have these outdated assumptions of, well, there are exposures we choose to have, there are exposures we don't choose to have, and we can only control those that are lifestyle factors. And we reject that, just as the President's Cancer panel did, that industrial chemicals are still an important part of cancer in much of the world, and we need to control cancer by controlling these as well as controlling those caused by cancer, tobacco, alcohol, diet, inactivity. There should be complementary strategies. Finally, gene-environment interactions. A lot of questions come up about genetic susceptibility. Some tumors are due to very rare mutations that are inherited, making some people more or less susceptible to cancer. But they're very rare. Sometimes it makes people very susceptible. But most the genetics we study, they make someone susceptible only if they're exposed. If we remove the exposure, the genetic variant by itself is not harmful. Those are so-called common variants rather than rare mutations. So the contributions of environment to cancer remains underestimated. We know it's a problem. It's a bigger problem in the rapidly industrialized countries of the world, and it spanned the gamut of, of traditional exposures we've known a lot about, combined with new chemical exposures. And the contributions of mixed exposures to cancer in both work and community still remain relatively poorly understood, and we need to leap in our exposure assessment tools to get a handle on this so we can prevent cancer rather than treat late-stage cancer. So for clinicians, be more aware, potential hazards, use the databases online, advise patients to reduce exposures to what they can control, smoking, diet, plastic containers, etc. So thank you. I will end there. So now it's my pleasure to introduce Dr. Monica Koliakovo. Dr. Koliakovo is a professor in the Department of Genetics at Harvard Medical School. She studies how environmental exposures impact biological mechanisms that are critical to reproductive health. She's one of the nation's leading researchers in cell division process of meiosis, which results in the formation of eggs and sperm. And she has a rapid screening process of how everyday chemicals affect DNA, and it illustrates the cost of an increasingly artificial environment and to equip the world with accurate information to enhance public awareness. Dr. Koliakovo is an associate editor in several key journals of the world, such as Genetics, PLOS Genetics, and she's a recipient of a number of prizes in science, the most recent, the Harold and Golden Lampert Research Award at Harvard Medical School in 2013. So we're very fortunate to have her speak to us today. Monica.
So I'd like to start by thanking both Angela Alberti as well as Dean Wild for the opportunity to be here and tell you a little bit about our work. And thanks to David for the introduction. So as you probably already had a feeling for in this introduction, there are various different chemicals in our environment. We're only starting to understand how they can impact health, and they can have various different kinds of effects on health. And so in my laboratory, what we're interested in is understanding the impact of these exposures on reproductive health. And that's why the title that I put up here says Germline Exposure to Our Chemical Landscape. We're assessing the effects and the mechanisms of function as a result of those exposures. And what I hope you can get a hint of from this title is that because the basis for reproduction, the formation of these reproductive cells, egg and sperm, are happening at a moment in time where it's ethically and technically impossible to look at this directly in humans, we rely on the use of model organisms to study this. The one that we're focused on in the lab are worms, and I will introduce you to this model organism in a minute. So, as I mentioned briefly in the title introduction, meiosis is this specialized cell division program that is the basis for reproduction. It's the cell division program that will allow you to form the egg and the sperm. And the reason why meiosis is different from a mitotic cell division is that at the end of the day, it results in the reduction of the number of chromosomes that you have by half. And the reason why you need this is so that the egg and the sperm, which now carry only half the number of chromosomes that other cells would have, when you undergo fertilization, as shown here, you will be able to reconstitute the right number of chromosomes in the new cell that is formed. But what we all know is that meiosis is an imperfect process. There are problems that can happen, there are errors that can happen, and that will result in the formation of cells that do not carry the right number of chromosomes. And that's what we refer to as aneuploidy. The consequences of aneuploidy, the formation of egg and sperm or embryos that don't carry the right number of chromosomes, can be severely deleterious for reproductive health. And so in the next couple of slides, I will introduce you to how serious this problem can be. If you look at the leading causes of infant deaths in the United States, exemplified here is the data for 2011 and 2012, what I hope you can appreciate is that the leading cause in the top here are congenital malformations. And that is far higher than something that we hear a lot more in the news, such as sudden infant death syndrome. It turns out that these congenital malformations are a result of problems, errors during meiosis. Not only that, 35% of all clinically diagnosed miscarriages, so this is probably an underestimate, are due to errors during that process of meiosis, as are 4% of all stillbirths and birth defects such as Down syndrome, trisomy of chromosome 21. And so it becomes very, very important to understand not only the genetic drivers of the process of meiosis, but also the environmental impact on this process. And that became very apparent to us because we were a basic genetics lab, right? We were identifying all these genes that are important for the process of meiosis, so trying to understand what drives this complex biological program. And then a few years ago, we realized we live also in a very complex environment. There are over 84,000 compounds that have already been produced. At least 1,000 additional compounds are added into the environment every year. And when you look into the literature and try to find out how many of these have been tested for an impact on reproductive health, it's really very few. So we really do not understand how these two are superimposed onto each other. We know that there are various different exposures, agents, environmental factors that have been correlated with an impact in reproductive health. For example, as mentioned in the introduction, use of chemotherapeutic agents, exposure to pesticides, plastics, all the way to maternal age. And so we came up with the following question in the lab, which is if we want to understand the environmental contributions to aneuploidy, to the formation of these egg and sperm that don't carry the right number of chromosomes, can we use this roundworm, it's a microscopic model organism called Caenorhabditis saligans, to investigate the impact of our chemical landscape on reproductive health? And here's a photo of the team, both past and present, that are currently engaged in this research in the lab. And so let me just remind you really quickly about what happens during meiosis, okay? So prior to entrance into meiosis, your genome is fully replicated. You duplicate all the DNA that's there. And then a series of challenges start to come up. First, now you have these two identical copies for every chromosome. They have to find each other and pair with the right partner. We know that also there is exchange of genetic information that takes place during meiosis. You basically swap bits of the DNA between these chromosomes. That is why meiosis provides for genetic diversity in the population. That's why I don't look the same as David, and David doesn't look like my tray, right? So that's why meiosis is so important. And as you proceed through meiosis, the chromosomes are fully zipped up as well by a scaffold that holds these chromosomes together. That zipper goes away. But now the chromosomes remain attached because of the swapping of that genetic information that took place, and now they're under tension. So when these microtubules that are like cables that attach to chromosomes and then pull them apart so that they can divide, when that happens, now these identical chromosomes are separated apart. That's the first meiotic cell division. But what makes meiosis very interesting is that you followed a single round of DNA replication by two subsequent uninterrupted rounds of cell division. So that now again, this genetic material, this blueprint, gets split in half through what's called the second meiotic division, and you end up with the egg and sperm that now carry only half the number of chromosomes, okay? So the question, and I hope you can appreciate from this simple cartoon, is this is not a trivial process. There are many different steps that can go wrong, right? And what we wanted to ask is, where can environmental exposures be affecting any of these steps during meiosis? The model organism that we use in the laboratory is this worm that I'm showing you here. When it's fully grown, it's about only 1 millimeter in length. And it offers a series of advantages for those of us who want to look at what happens in the germline. The first one I hope we can appreciate from this movie, which is that it is a transparent organism. We can look at everything that's taking place inside this worm, and with the advent of things like green fluorescent protein and so on, we can actually look at specific tissues in real time and follow what's happening. This system also shares, believe it or not, a tremendous amount of gene conservation with humans. It turns out that anywhere between 60 to 80% of all the genes in the worm are also present in humans, right? And what I hope you can appreciate from this cartoon is that 50% of all the cells in the worm are contained in this structure that I highlighted in blue, which is the germline. So for those of us who want to look at what happens in meiosis, 50% of everything in this worm is undergoing the process that we want to focus on. If you dissect this worm, if you cut it here and you isolate only one of these two gonad arms, and you now stain chromosomes so that you can look, imagine that this is a tube. It actually is. The nuclei are lined on the walls of this tube, and they're moving. And what I hope you can see is everything that's in white here represent chromosomes in these nuclei, and here I'm showing you higher magnifications. You see a time course of progression. You can see changes as you proceed through the process of meiosis. And chromosomes organize within these nuclei in very characteristic ways. And I should add, these changes in how chromosomes are organized, they are the same whether you're looking in yeast, in worms, in flies, or in humans. But what this allows us to do is easily identify situations in which you can perturb this process, where you've either affected the timing with which things are progressing or the actual organization that these chromosomes acquire. And we use various different markers and reagents to try to understand where there might have been a problem. So when we started with this in the laboratory, we chose to focus on a very highly prevalent chemical that's in the environment. And our compound of choice was bisphenol A, or BPA, which many of you must have heard a lot of. It's highly prevalent in the environment. It's a very commonly used plasticizer. It's in the inner lining of cans because it protects the product in the can from basically damaging the metal container. It's present in dental sealants. It's present in fabrics. So we're exposed to it every day. It actually exhibits an exergenic activity. It's an endocrine disruptor, right? But it turned out that studies that initially came from Japan showed that there was an association between the risk for miscarriages with women who had a very high level of bisphenol A either in their blood or their urine. And when they analyzed the chromosomes for the miscarried fetuses, what they found was that the number of chromosomes were not normal. So remember, that's aneuploidy. So they started to make a correlation between perhaps high levels of bisphenol A can result in a higher probability of miscarriage. And it was years later that very elegant work done in a model organism, in this case the mice, that they showed that indeed exposure to bisphenol A can directly affect that very basic process of meiosis, resulting in problems with how those chromosomes are fully aligned by that scaffold, the synapses, resulting in problems in aberrant morphology, shape, and organization of the chromosomes, altering the way that genetic swap of information, the recombination process takes place, and ultimately not allowing for an accurate partitioning of the chromosomes when the cells divide. So here's an image from one of these papers that shows you chromosomes are in red, those cables that are pulling them apart, the microtubules, are in green, and instead of having all the chromosomes aligned right here, which is where they should be, what you would see after bisphenol A exposure is that some of these chromosomes were not at the right position. And then subsequently, when you start repartitioning them, you're not going to distribute them properly. So this created a very interesting situation for us, which is, given that we already knew what was happening in the mammalian system, right, could we identify whether bisphenol A caused a similar kind of effect in our model organism? But the second question was, can we take it a step further, and perhaps because our system is so easy to manipulate, can we identify the mechanism by which bisphenol A is causing these problems? So can we identify the meiotic stages and processes that are affected, as well as the genes and the pathways that are altered? Where is bisphenol A going to be working in this germline? And I'm going to just really succinctly elaborate on some of the key findings, which is that at a very high concentration of exposure, 1 millimolar, this was the equivalent of an intake of 2 parts per million. So what this was within the range of the mouse studies that I just showed you before. It was within the range of people who, because of occupational hazards, are exposed to high levels of plasticizers or who manipulate cash register receipts, which are coated with bisphenol A. So at that level of high exposure, what we saw was a dramatic effect on the fertility of these worms. So now they were unable to lay normal numbers of eggs. There was a lot of sterility. And we also saw that the few eggs that were laid, very, very few were able to hatch. And all the eggs that hatched, those larvae never progressed into adults. So the impact of bisphenol A exposure was very severe. And it resulted in exactly the same phenotypes, the same defects that had been reported in the mouse model studies. We saw problems with the ability of how things were getting recombined, the ability to repair DNA damage. We saw problems with the quality of how these chromosomes look like. We saw the fragmentations, the aggregations. But in addition to that, when we looked at the first embryonic cell division, and here's an example of what happens if you expose these worms only to ethanol, which is what we were using to dissolve the bisphenol A, and what happens if you expose them to bisphenol A. Again, in green, what you're seeing are those cables that are going to pull the chromosomes apart. In red are the chromosomes. Instead of nicely aligning at this central position, as it does in the control, chromosomes never fully congressed. They didn't quite align properly, but more importantly, and I hope you can see it indicated by the yellow arrow, we started to see these bridges, these connections that had never been resolved between the chromosomes. And you can imagine that as these masses are being pulled away from each other, if they're interlocked, you're going to mechanically damage them, okay? And so this also explained a lot of the embryonic lethality that we were seeing. We also saw formation of abnormal spindles, which are these structures at the tips here, and ultimately these cells during mitosis were not capable of fully dividing, okay? So we were detecting problems in meiosis that were getting carried all the way into a mitotic cell division and affecting embryogenesis. But what was very critical about these studies is that we also found out that there was a conserved set of genes. There were specifically 3 to 4 genes that are conserved and present all the way to humans and that are very, very important for DNA repair that were not being expressed normally following bisphenol A exposure. This was the first time that this was reported. A few years after that, several mammalian studies reported the same finding, which is that bisphenol A exposure is causing many of these defects that I just described because you're deregulating how certain key genes that are important for repair to be present at the right time at the right level. Okay, their products are not there at the right time at the right level. So that also meant that we could use this model organism to really tease apart the way in which many of these chemicals might be impacting reproductive health. And so one of the things that we got interested in the lab is, okay, we can identify a chemical that might affect reproduction. We can understand the mechanism by which it's doing this. Can we go bigger than this? Can we start looking at hundreds of chemicals and really, in a very quick manner, take advantage of the fact that this worm goes from an egg to an adult in just 3 days, and then it lays 300 eggs. A single worm will lay 300 eggs. That set of eggs will only take 3 days to become an adult again. So if you think about the life cycle, it means that we can look at multiple generations in a very, very short period of time, and it's a very amenable genetic system because you have hundreds of offspring that you can look at. You have the power of numbers as well in looking at an effect. Can we harness that? And this is a cost-effective system. We keep these worms in Petri dishes in a regular incubator. It is highly predictive of mammalian reproductive toxicity based on several other studies that I'm not going to go into details today. Can we use this to screen hundreds of other chemicals and identify those that might be impacting reproductive health? And one way in which we've been doing this in the lab is taking advantage of another interesting feature of this model organism, which is C. elegans exists in 2 flavors. They're either hermaphrodites, meaning they produce both egg and sperm and can self-fertilize. They carry 2 copies of the X chromosome. Or they can be males, which carry just a single X chromosome. A self-fertilizing hermaphrodite, most of the time, will only lay hermaphroditic progeny. Okay, males are very, very rare in the population. Less than 0.2% of those eggs are destined to become a male. And that's because, if you think about it, to become a male, it means you carry a single X. It means there must have been an error in how chromosomes were partitioned. It means that you generated either an oocyte or a sperm that simply did not have an X chromosome, And when it matched with, let's say it was an oocyte that had no X chromosome and it got fertilized by a sperm that carried us an X, that's how you recover the single X. So males are very, very rare. But we know that if you affect meiosis, you start to randomly have problems, right? You start to randomly partition these chromosomes. The frequency with which you might have errors in how that X chromosome gets partitioned goes up. And in fact, if you look at meiotic mutations, we now start to see 30, 40% males in the population versus 0.2%. So it's very easy to detect something that's messing up with meiosis. So we could use this, what is called a high incidence of males output or phenotype, as a readout to understand what chemicals might be causing problems during meiosis. And that became even easier because we could actually take advantage of the following. Barbara Meyer's lab at UC Berkeley had identified something called the XOL1, XOL1, which is a male-specific promoter. It means it only drives expression of a gene in males. She fused it to the green fluorescent protein. Anne Villeneuve at Stanford University then used this construct, what we call a reporter construct, in a very clever way. She thought, if I mutagenize worms, right, to identify actual genetic mutants that might be important for meiosis, and I have the worms carrying this reporter construct, if I start to see more males in the uterus, they will glow green. I know that those eggs are destined to become a male. I must have hit something that's very important for meiosis because males are rare. So can I see these green eggs in the uterus? And it turns out that they're very, very obvious. This is a snapshot of what that uterus looks like. You can really see these green eggs, and they would normally never be there, right? So we then decided, well, we can use this strategy in a slightly different way. What if we now use that same reporter construct and expose our worms to chemicals? We can do this with multi-well plates, so we have various different chemicals, various different concentrations. They're swimming in this liquid that has whatever toxic agent we want to test. Can we then see if we see these green eggs? And to make this even a more robust system, what we did is we introduced this genetic mutation on a collagen gene called COL121. And the reason we did that was to break down the cuticle barrier of the worm so that we can really now introduce very, very low concentrations of the chemicals. For example, bisphenol A, we can significantly dial down the concentration. It's no longer 1 millimolar, it's 100 micromolar, it's very, very low. Now we can work within the range of what's used in cell culture experiments and all these other systems. Can we see an effect? So can we see these green eggs? But to make it high throughput, we can take advantage of still another technological advance, which is that there now is a machine that allows you to FACS sort your worms based on fluorescence. So you put these live worms through this machine, and it will detect whether you've got green eggs in the uterus or not, and how many. So what's the fold increase compared to, for example, the vehicle alone? And so we've been successfully applying this strategy, and I'm just going to quickly show you a slide that exemplifies the kind of library that we've been investigating now in the lab. We've been looking at components that are used in hydraulic fracturing, components that are used in the processing of crude oil, various different pesticides and antimicrobials, phthalates, which are highly prevalent in the environment because of plastics, cosmetics, what you spray on crops, shower curtains. And it's been working very successfully. We've been able to identify which of these are causing problems in reproductive health, and now we're starting to investigate why they're causing such problems. Because that's the final step. We want to understand the pathway, the mechanism by which they're affecting this, and ultimately we want to find out chemicals that might exhibit transgenerational effects. So a single exposure of mom that can lead to effects not only in the children and the grandchildren, but in the great-grandchildren as well. So with that, if there's one important take-home message that I want to leave you with, is that we can start to understand very important steps of this complex biological program by taking advantage of basic model organisms. This can inform what happens in the human scenario. Questions that are raised in humans can be answered by what happens in the worm, so these can feed back into each other. This is not perfect. You need a combination of different systems to ultimately get a full picture of what might be happening. But we hope that this is one element in a cascade of things that might be used in assessing what chemicals might be doing before they're delivered into the environment. And with that, I'll thank the lab as well as funding sources. Thank you.
Thank you, Monica. Our last speaker before questions is Dr. Maitri Mazumdar. Dr. Mazumdar is an assistant professor of neurology at the Harvard Medical School and Boston Children's Hospital, where she's an assistant professor of pediatric neurology. And also, she's an assistant professor in the Department of Environmental Health at the Harvard T.H. Chan School of Public Health. Her research focuses on environmental exposures to compounds like arsenic and neural tube defects like spina bifida. Her studies take place in Bangladesh, where there's an estimated 70 million people who've been affected by arsenic-tainted drinking water. In 2016, Dr. Mazumdar was the recipient of the Outstanding New Environmental Science Award from National Institutes of Health. Thank you.
All right, this is quite a thrill. Thank you for inviting me to participate in this. And I have to say, this is great. You can't hear me? Is the mic on now? Now it's on? Okay. Maybe it's too far away. Well, I want to get it right. Because what's the point, right, if you can't hear? All right, is it better now? Okay. So I'm a pediatric neurologist, and I've come to talk to you about children and environmental chemicals. And I'll just give you a quick overview about what I'm going to cover. We'll spend a little bit of time talking about why kids might be more vulnerable to environmental chemicals. I'll use lead poisoning as a paradigm to discuss developmental neurotoxicity. Lead poisoning's in the news these days, and I think in addition to being timely topic, it's one that really has been studied well in Boston in particular. And then end with just a plea, as David has already mentioned, about a precautionary principle in our approach to environmental chemicals. So why are kids different, or why might be kids different, or why might kids be more vulnerable to the things that we're talking about? And I think kids have a number of different things that put them at higher risk. They have different and unique exposures, which we'll talk about, different from adults. They have a physiology that's very dynamic. They're growing, and their growth provides windows where they may be more susceptible to harmful chemicals in the environment. They live longer, so they have a longer chance to develop problems, even from an exposure early in life, and we'll talk a little bit about that. But I think most importantly, they rely on us, they rely on adults to protect them. They can't make a lot of the choices about where they live, or their lifestyle, or what they eat, or what they're exposed to, or what's regulated in their environment. They don't vote. And so they are vulnerable just because of their situation or their position in our society. So let's talk a little bit about different and unique exposures. So in addition to breathing the air and eating food and drinking the water, children are exposed to unique exposure pathways. They get chemicals pass through them through the placenta, through an umbilical cord, which is can be a very protective environment. The placenta often does shield out a lot of chemicals, but it's not perfect. They are exposed to chemicals through breastfeeding, which is just a very different way of getting exposure to a chemical in the environment. They have exploratory behaviors that are different from adults. They put things in their mouths, a toy that maybe, like a train toy that we might have on our desk, my son puts in his mouth. I mean, what can you do about that? So they are different. They have different stature and microenvironments. They're shorter and closer to the ground, so they're closer to the carpets that might have chemicals. They're just there, right? We're up here, they're there. They breathe different air because they're shorter, right? What comes to them into their respiratory passages may be different from what comes there. And they don't understand danger. right? If you say, don't go over there, it's dangerous, that will be the first place that they go, right? So children are different, and you have to think about them differently when you're designing strategies to protect children. So some of these I've already talked about. So they're breathing zones. Children are different within themselves. A crawling child might be more at risk than a child that can be put in a crib, unless the crib itself is dangerous. Like I said before, they're closer to the carpet. They also breathe more air per surface area, right? So for their weight, for their build, they're taking in more. They have a higher intake of food for their weight, and they eat different things, right? So they have a different diet. They have milk maybe more. They may have different types of things that are in their diet. Okay. I'm a pediatric neurologist, so when I talk about children, I talk about children's brains. And it's just a model of an organ that is undergoing development, that's growing very, very rapidly, all the way from the in utero period through adolescence. And as Monica mentioned when she walked us through the steps of meiosis and gamete formation, brain development is also a very, very complicated process that has many places where things could go wrong. So just to talk about them or to highlight them, for the brain to develop, it has to do many things in sequence in a very programmed, very predictable way that involves many different things, including genetic influences, nutrition, but also can be disrupted by chemicals or other things in the environment. So just to walk through them a little bit, see if this works. Proliferation is the first step. The cells in the brain have to multiply in number. Then the cells, once they multiply, they have to travel to where they're going. So that's a really exciting part of brain development to study and to watch, to see the brains kind of form from the inside out. They start in the middle of the brain, but then have to make their way up to the ends of the brain. They form these connections, and then there's which is called synaptogenesis. And then there's also a very predictable trimming of connections as well, which is a natural part of brain development. It's called pruning. Within that, there's a very sophisticated formation of transmitters and receptors, and all of these things can be influenced by substances in the environment, by experiences in the environment as well. So children develop on a very predictable course and in sequence. And so this is a slide from Chuck Nelson's group, who's at Boston Children's, showing that within the first year of life, there are development of vision and hearing pathways, followed by language, followed then by higher cognitive function. And this is all related to brain development and this pathway, these series of steps that occur within the first 15 or so years of life. Okay, so why is that important to what we're talking about? Well, the reason I went through that is because I wanted to make the point that what happens to the brain of a person who's exposed to a chemical can depend on where in that process they are when they're exposed to the chemical. So this is a cartoon from pathology, from brain cuttings, of a population of Japanese people who were exposed to mercury through dumping of industrial waste into a water source in Minamata, which is an area in Japan in the 1950s. And if you looked, and these are their brains, or cartoons of their brains in cross-section, and if you look at the adults who are here, children who were not, who were born after the poisoning incidents, and the brains of children who were in utero, whose mothers were pregnant at the time of this poisoning. They were all exposed to it through drinking of water, though this population was exposed transplacentally or in utero, you can see that their pattern of injury is different. And so these dots are meant to represent areas of injury. So in adults, the damage was more focal, more restricted to certain parts of the brain. But in those who were exposed prenatally, or while they were in utero, the damage was very much more widespread. Right? So just the age and the developmental stage that these individuals were at the time of exposure affected how they responded to this exposure. Okay. All right, so that's an overview of developmental windows of toxicity, or why I think children may be more susceptible. And now let's talk about lead. And once again, there are many chemicals in the environment that we could talk about, but lead is really the paradigm, and also it's very timely. So right now, because of the situation in Flint, Michigan, we're learning a lot about lead in the water and lead in water pipes. But it's important to remember that lead is really everywhere in the United States, and there are multiple sources of exposure for children, even though it's improved substantially over the past decades. So in Flint, as many of you know, there is lead in the water that it's related to leaded pipes. In Boston, the main source of lead exposure is lead in paint from houses that were used lead paint prior to the ban of lead in the 1970s. And then every so often there's a story about lead in paint from toys that are imported into the US. Prior to the removal of lead from gasoline, lead in the air from gasoline emissions was really the major source. Okay. And elevated blood levels affect the brain, and we'll talk about that to some extent, but really affects every part of the body. At high levels, so levels at about 100 or 150 micrograms per deciliter, children used to die from lead exposure and still do in many parts of the world, such as Nigeria, where children are exposed to lead in lead mines. We'll talk a little bit about developmental toxicity and the studies that were done here in Boston, where lead was shown to be associated with decreased IQ, decreased hearing, decreased growth, and other maybe less dramatic, but still substantial neurologic outcomes. Actually, even though we're talking about lead and the story of lead is really one of our biggest public health successes. Blood lead concentrations in the US have reduced dramatically over the past decades. In the 1970s and 1980s, the median blood concentration among children, median lead blood concentration was around 15, which is a level that would be considered completely unacceptable by today's standards. And now it's less than 2. And this has to do with restrictions on the use of lead-based paint and also the phasing out of leaded gasoline. But let's talk about what lead does, and we'll talk a little bit about studies that were done here at Boston Children's Hospital and just on the street. So in the 1980s, one of the very first studies done to look at the effects of childhood lead exposure showed that if you looked at the teeth of children who were exposed to lead, and you looked at children who had high levels of lead in their teeth by some measure, and those that had low, the IQ, the verbal IQ scores of the group with lower lead levels were higher, right? So there was a drop in IQ scores among children who had higher levels of lead in their teeth being a measure of chronic exposure. The number of IQ points wasn't very high. That change, it was around four IQ points, which may not seem like a big change. But if you look over a population, over a big period of time, these children who were exposed to lead consistently were doing a little bit worse than their peers who were not exposed to lead. This started a number of studies tens and 20s of studies all over the world to look at lead and IQ. And you can see that the results were really very varied with different estimates of the effect, different shapes of their curves, different ranges, both of IQ and of blood level. But when you sort of combine the data and look at them together, you can see that there's a pattern that at lower blood level concentrations, there's a steep drop in IQ, with this being sort of a range of IQ. And as you go out, the shape of this curve flattens out. So there's certainly effects of lead that are seen throughout this range of exposure. But at the beginning, at these lower levels, is where maybe a little bit of lead is the most harmful. This has led, this and these public health interventions that I mentioned before, has led to a drop in the level of lead in the blood that we consider to be harmful. Before, we used to say that a level over 60 was harmful, and under that was probably okay. And then that threshold has come down over the past number of decades. To when I was in medical school, it was 25 and then reduced to 10. And now, as we understand that lower and lower levels of lead can still cause this drop in IQ or other measures of neurologic function, we don't even call it a threshold anymore. We say it's a reference level, that there's no level of lead that's okay. And this level of 5, which is the level that was used in Flint to identify children with high levels, is still something that we're trying to get lower and lower and lower every year. So those are the effects on children, but are there effects that last longer, or how long do these effects last? So there's certainly a hypothesis that's out there that children who were exposed to lead or exposure to chemicals early in life has not only effects on the number of cells, or you can use this as cognitive function, or a number of different outcomes on this y-axis. Not only do people who are exposed to this chemical exposure early in life, so this is, let's say, prenatally, and then this is their lifespan. This would be normal development. There's some cognitive function and then some normal decline. I've taken the ages off here because I'm reaching the point of inflection and don't really want to acknowledge that. But let's say you're exposed here, and then there's a measurable change in your performance on tests because of your exposure to lead. So this has been shown, but there's a hypothesis that it also causes a decline, a more rapid decline as your age goes on. Now, how would you test that, though, in people? So in our studies, the ones that I mentioned briefly, you can look at kids and sort of get some estimate of their early life exposure, either through biomarkers or teeth or some other thing. But if you're, let's say, 70 or 60, how do you estimate what this exposure is? It's really hard to do. This is a hard hypothesis to test. So this is where we often go to animal models because, like worms, they have a shorter lifespan. And so there's this very interesting study that's out there in the animal literature, and this is from mice, and this is a group in Rhode Island, that gave lead to mice early in life. So this is their lifespan. And these are controls. So these are mice that were treated with lead, and these were controls. And they're treated with lead, and they follow them through their lifespan, which is about 2 years. And as they got older, their expression of genes that are involved in protein folding, genes that are involved in processes that are similar to those of Alzheimer's disease, expression of those genes went up. And then later, if you looked at the neuropathology of these animals, you could see that they had more formation of these plaques and accumulations, I guess, there were more in those that were treated with lead than those without. So the hypothesis from the animal literature is that early life lead exposure may be a risk for later life Alzheimer's disease. So once again, it's hard to test that in humans. Let's say I decided to start a cohort now, or when I was 30, and then follow them for 70 years. By the time it comes the time to evaluate them, they're 70 and I'm 100, right? So you can't really do very much about that. And so we went back to our cohorts of children who were born in the 70s and 80s, and then we tried to see what they were doing. And I'll run through this quickly. We saw that we were able to get about 80 kids, 80 adults, 30-year-old adults, who had been given, who we had information about their umbilical cord lead. And we saw that they still had decrements in their IQ when compared to those who were not exposed to lead. And we also saw, using a biomarker for Alzheimer's disease, where lower levels is associated with a higher risk of Alzheimer's disease, we found that, let's just use this one, that those who had higher cord blood lead concentrations had lower amounts, or at least nothing in this area, of these particular proteins. Okay, so it's an interesting hypothesis. It's one of many, but it's there. So what we think we know, I'll just end now, we know a lot about lead, but we really don't know much about these other chemicals. And there are many chemicals in the environment. Lead is a paradigm because of its effects on the brain, because we have a lot of literature about it. But there still are these other things that we can use a similar approach to study. And as David mentioned, we should apply a precautionary principle and not wait to demonstrate that these things cause harm, or to see that they cause harm, but especially in the case of children, err on the side of caution. So our conclusion is that children have heightened vulnerability because of their developmental processes, because of their position in society, and that lead poisoning illustrates the developmental toxicity. There's increasing recognition of effects at low levels. The effects are long-term, and there may be latent effects, such as an increased risk of Alzheimer's disease, and that we should apply a precautionary principle for the introduction of chemicals into the environment. And that's it.
So we go for 20 minutes, right? Oh, yeah, he's gone. Okay, good. So I'll go through as many as we can. Thank you, my chair. We're going to give people about 30 seconds to clear the room, and then we'll start with questions. We thank you for your questions. We have quite a few very interesting questions that were written down. Excellent questions. Okay, good. So we'll start. Okay, so we'll start with questions. The first two for Monica Koliakovo. One is, since human oocytes are generated early in development, at what period do exposures have their main effects? And for example, does a mother exposed to BPA have a risk of offspring with aneuploidy, or do her children?
So the process of meiosis is happening inside that developing fetus. So if the mother gets exposed, right, what's happening is you're affecting the meiosis that's taking place inside that baby. So ultimately what you're doing is you're affecting the quality, for example, of the eggs that child will eventually produce. The impact is going to be on the grandchild, right? So that child might have a higher incidence of aneuploidy because it got exposed while it was in utero. Having said that, meiosis is a process that takes decades. It's initiated in that developing fetus, it's then arrested, and then later, as the child reaches puberty, that's when you start to complete meiosis. So you're in what's called diapause. You're arrested at a stage called diakinesis, and then you resume meiosis and you complete it, which means that additional exposures that can take place, even in that now adolescent woman, for example, can then impact the subsequent and tail steps of meiosis as well. So it's not limited to what happens in utero. You can have additional exposures in your lifetime that can affect things later as well.
Thank you, ma'am. So one more, Monica, for you. There are actually many in there about chemicals, but what about those of us over 50, outside of our reproductive window? Do we have to worry about plastics? Should we throw out all our plastic containers?
Right. So I focused a lot on what happens in terms of reproductive health, but yes, you need to be aware of the fact that exposure to this plasticizer and plastics in general can have various different deleterious effects. The exposure to bisphenol A has been associated with cardiovascular disease, with diabetes, with it possibly being what we call an obesogen. It can be affecting the ability to gain weight, right, and be associated with the obesity epidemic in the United States and other developing countries. So a couple of things that people should be constantly aware of is you do not want to heat plastic containers, do not microwave anything that has plastics. You do not want to, if you can, put anything that's plastic in a dishwasher because of the high temperature that that will achieve. So you wash things by hand, and you have to use very, very gentle detergents and try to use the non-abrasive side of a sponge, for example, when you're cleaning the container, because you don't want to facilitate leaching out from that plastic into whatever you're going to put in that container later. So that's something that you need to be very consistently aware of, because the impact is not only about reproduction, right? And in the case of men, we're beyond where spermatogenesis is happening all the time. This is an issue no matter what, even in the context of reproduction in older men. So in my household, I've basically eliminated as much of plastics as I can, and I'm very, very cautious about what I do or do not put in contact with plastics.
Thank you. Another question, can being exposed, not wearing a covering to protect you to X-rays be harmful to you? The answer is yes. You should always be protected with lead aprons, or the equivalent, basically lead aprons when you're being X-rayed. It's cumulative, however. A single time it happens is unlikely to increase your risk a lot. It's cumulative over many years. Another cancer question. You mentioned a lot of carcinogenic substances. Which single one should be targeted first? The obvious, smoking. Well, actually, it's a good question. You can go onto the EPA website, Environmental Protection Agency website, and look at carcinogens, which is similar to the International Agency for Research on Cancer. There are about, in the group that's 1, known human carcinogens, and 2A, probable human carcinogens, the total is about 400 chemicals. Definitely 1 and 2A should be avoided, right? Then there's possible, and then there's unknown. So I think the list is longer than you'd expect, beyond just things like smoking and a few others I mentioned, but most of them are not necessarily in everyday use. Okay, that's why the EPA site explains it a little better. Another question. Let's get a lead question from Maitri there. We dealt with microwaving this. Literally everything is made up of chemicals. How do you deal with self-proclaimed scientists in social media who say that any chemical that you can't pronounce is dangerous? Actually, it's a good question. In a way, and I'll see what Maitri and Monica feel about this, to say that everything causes cancer, everything causes brain damage, everything causes reproduction, has a long name, is disempowering. It's not empowering. We really want to know a couple things. One is not every chemical is harmful. Of the 80,000 in the market, I'm fully prepared to realize or understand that the majority probably do not cause cancer or any serious effects, but we just need to know. That's the one thing. We have a right to know what does and doesn't cause. Realizing perhaps the majority do not cause any problems. Therefore, you focus on the ones that are the problems and become empowered to minimize or eliminate those exposures. So when you think about it, if you say, well, everything causes disease, that's very disempowering, because what are we supposed to do? So the facts, that's why we do scientific research, to prioritize those things. All right, Maitri, you're not off the hook. Let me see. Water safety. Is it safe to drink tap water in the U.S.? Is filtered tap water better than unfiltered? What about boiled tap water? Boiled like boiling it, like they do in much of the world.
Well, certainly tap water is in the news, and so I think I might need help from David. But I think it depends on the region where you are, and I think this is an example of how different areas and different municipalities handle things differently. So I think in many communities, the water that comes from the pipes and tap waters is probably very safe, and is well treated and clean and without significant contamination with chemicals. But it is very area dependent.
Yeah, I agree. And also, within an area, there's going to be some issues. Boston, we're fortunate, the Quabbin Reservoir is a very good water supply. However, we have conduits in Boston that are very old, pipes, and can be leaded. So the water supply is actually outstanding. But when it gets to the house, if you have lead pipes, conduits in the house, or aging lead pipes, then that house can have a problem, or the group of houses. So I agree with Moitri. It's regional, but it's also very local. And so it is worth thinking about looking at your locale, what's been known about it, and what testing you may have to do. Finally, boiling water is done in a lot of the world to avoid infectious contaminants. We never really recommend that. So think about high school chemistry. If you're worried about metals and you boil the water, what happens? You concentrate the metals, all right? Because you're boiling off steam. You're letting those nice water molecules go away, and you're concentrating lead, arsenic, or manganese. So there's no reason to boil it in the U.S., although there may be situations where the water is not safe. And the other thing is bottled water. There was a study done some 20 years ago of some of the popular bottled waters around, and they had higher levels of contaminants than the municipal water supplies in places like New York and Boston. So certainly be aware of it. Now with the internet, you can get a lot of information on your local water supply. Okay, next question. Series of questions. Quick. Is secondhand smoke still a problem? Yes, but it's declined a lot because of regulatory and educational efforts. Secondhand smoke, a spouse living with a smoking spouse has about a 30% increased risk of getting lung cancer. So your spouse is the number one target. But in terms of the workplace and the general environment, there has been in the U.S., I think, really significant progress in this. Okay. Let's see. PCBs. There's a question about PCBs in a site in Hyde Park. The company wants to put them underground and vaporize them. They probably want to incinerate them is what it is, which is what happens sometimes at Superfund sites. That sounds weird, but sometimes high temperature burn for certain organic chemicals is actually one of the ways of remediating it. It's not that they just take the chemical, it's the soil's contaminated, and so they basically burn the entire several feet or whatever the hydrogeology and the environmental engineers say you need to go down to get to clean soil. And so that is still an acceptable environmental engineering procedure, realizing that the controls on their emissions are have to be quite strict and latest technology. Lead in chocolate and arsenic in bottled water. Moitri?
So I don't know about lead in chocolate. I think the issue about lead in food products, however, is certainly one to talk about. In the U.S., food and food products, or at least when it comes to metals, are quite well regulated, or at least that's my opinion. In the rest of the world, in the rest of the developing world especially, lead is used as something to brighten the colors, to increase the weight for spices and other things that are sold by the weight. And so lead contamination of food products and spices is a very big issue. David and I were both involved in a study not very long ago where the U.S. had recalled batches of turmeric that had been shipped to the United States because their own testing, so they do testing fairly regularly of imports, had shown very high levels of lead in these food products. And in the sites where we work in Bangladesh, we found that we had high levels of lead among the children, and we didn't really know where it was coming from. And we went to their homes and we tested a number of things, including their turmeric, and we found astronomically high levels of lead, which has no role except it was probably part of a dye to make this orange spice more orange, because there was no reason to think that it would be in the ground or the soil. So whereas I don't think it's a big issue in the United States, I think contamination by metals and other chemicals in the food by lead is increasingly recognized as a health hazard to children.
Great. Another one for you, Rachel. How does medication that affects using for attention deficit disorder affect children's brain long term?
Attention deficit disorder is a particular diagnosis, one also that has a body of literature relating it to lead exposure, early life lead exposure. And so the way that, which is an aside and not really the answer to this particular question, so the medicines that are used to treat attention deficit disorder generally are a class of medications called stimulants, not unlike the coffee and the soda that we as adults use to increase our concentration. The long-term effects on children, one, they're short-acting drugs, so they don't accumulate in the body like some of these other chemicals do. But the long-term effects of these medications are related to the side effects of the medication, which include decreased appetite. So when we think about what the long-term effect of these medications are, it's more to do with reduced appetite and reduced food intake relating to slower growth, which has been shown to reverse when you take the medications away and kids get hungrier again and then eat again. So I think in terms of accumulating damage, there's not evidence for that in the literature.
Great. Okay, a couple more cancer questions. What, if any, toxins contribute to skin cancer, melanoma? Actually, it's not so much toxins, except, well, arsenic can cause skin cancer. And there are some hotspots in the country, New Hampshire, water supply, and some other parts of the Southwest. But mainly, skin cancer is an ionizing radiation, ultraviolet light problem here. So that means sunbathing, tanning booths. Really, I think tanning booths should be banned, frankly. Should we limit dental x-rays and MRIs? X-rays, medical x-rays should be limited, as we mentioned, to the extent possible. You need them, you get them, but you have your thyroid and other parts of the body covered with a lead apron. MRIs, contrast, do not emit radiation, any ionizing radiation, so there's not a cancer risk for MRI. Why do children have a high risk of leukemia? Well, actually, I might have given you a misimpression. It's not that they have a high incidence. It's actually, these tumors are rare in children. It's just when they get it, they're grouped into these big categories, and leukemia is one, and it's probably a disordered immune system from pre-birth. And so it's not really that they have a high incidence. It's just that when they do get cancer, they tend to group into these categories, leukemia being a big one and central nervous system being another. Okay, let's see. How can we prevent these chemicals from ending up in our products? So that's a policy question, right? So we're scientists here. We're trying to provide the knowledge base for which that should then be translated into action. Some of that action is policy on the part of government to regulate these things. So that they don't get into consumer products, into our homes and workplaces. And the other part of translating scientific information is to have the public be informed, because sometimes these chemical compounds can't be totally eliminated, but need to be avoided. So I think it's a great question, and it differs a lot where you live. The European Union tends to be much tougher using a precautionary principle than we are in terms of the amount of evidence needed to ban a compound. It's actually quite hard to ban compounds in the US, but that's why we do this work. Let's see, we got a couple more minutes. I've observed that people who are heavy smokers rarely live beyond the age of 60. Yeah, not much more can be said about that. I think that's a very astute comment. It's a sad one. and it's obvious what needs to be done to control that. The good news about that, by the way, so you say you smoked for 20 years, and you say, okay, it's too late, I'm already too far gone. Not true, okay? You stop smoking, your risk of lung cancer goes down 50% over the next 2 years. So when someone says that, you say, well, you're really a fatalist, because you still can do something about this. If a person has a blood transfusion, can they get cancer? No. Serious question, and it's actually no is the answer, because you usually get red blood cells as blood has been treated. Cancer cells, even circulating cancer cells, occur in very low frequency in the blood of patients with cancer, although they do occur, but it's not a transmissible disease. So that at least theoretically, even if one cell got through, your immune system would take care of it. It's a very good question, but we're not talking about infection. All right, so I see the red light. I think we got through as many of these as possible. Thank you all so much for coming.